Thank you so much, and I would like to thank the organizers for arranging this platform where we bring the science, innovation, and the shared purpose of patient care together. We at Alkeus aim to preserve the vision of those who are affected by retinal diseases. Our initial focus is Stargardt disease, an area of very high unmet need. Gildaretinol is a new molecular entity, which is one oral capsule once a day. It has been designed to reduce the formation of the toxic vitamin A dimers in the retina, which are the main culprit of retinal degeneration. Systemically, it is metabolized very similar to vitamin A. Gildaretinol has breakthrough therapy, orphan indication, and rare pediatric disease designation. Although rare, Stargardt disease is the most common form of juvenile macular dystrophy. 95% of the cases are due to ABCA4 pathogenic variants, which leads to the defect in the ABCA4 transport protein, leading to the lack of clearance of transretinal. If symptomatology presents at an earlier age, patients rapidly progress to legal blindness. What is the underlying mechanism of disease? With the defect in the ABCA transporter protein, transretinal accumulates in the photoreceptors because of the lack of clearance, which eventually forms bisretinoids. Bisretinoids further hydrolyzed in RPE to form A2E. The A2E in turn combine with other bisretinoids and their derivatives, as well as oxidized lipids, to form lipofixine. The accumulation of lipofixine caused the upregulation of complement pathway, increased oxidative stress, as well as cause the inflammation leading to retinal cell death. If you look at on this slide, both on the left side in vitro and on the right side in vivo, gildaretinol has shown a profound reduction of 70 to 80% in A2E formation. Currently, gildaretinol is currently being studied in two disease conditions, Stargardt disease as well as GA due to AMD. On the left-hand side, 4 boxes, you will see there is a comprehensive TEASE program of Stargardt disease across the whole spectrum of disease, where study TEASE 1, 2, and 3. Once the patients complete, they transition to TEASE 4. And I will go one by one on each of these studies. Here is TEASE 1. It was a randomized placebo-controlled study in patients who had at baseline well-defined retinal lesion. 50 patients were randomized to either high dose, low dose, or the placebo arm. At 1 year, patients from the high dose were transitioned to low dose as per the recommendation of the DMC, as no difference was found between the replacement of native plasma vitamin A level. The primary endpoint was the square root of atrophic lesion growth. Here are the results. Study met its primary endpoint, showing a 21.6 annual reduction in the lesion growth. And the analysis of the sensitivity analysis of the primary endpoint showed a 30% reduction when the non-transformed data was used. And also, without the supplementation of the placebo arm with the natural history data, the results remained significant. The drug was well tolerated in this population, and the adverse events were similarly distributed across both. We did not see any side effects or adverse events of vitamin A deprivation to the visual cycle, such as delayed dark adaptation, chromoptosia, and also night blindness. One case of papilledema was seen, which upon discontinuation of the drug temporarily was cured, and drug was reinitiated, and the patient successfully completed the trial. TEASE-3 is an open-label study which is aimed to enroll 20 patients. Thus far, we have enrolled 7 patients. 5 of them have completed the study. It is in a very early Stargardt disease patients, those patients who are basically diagnosed because of their family members have been diagnosed. It's aimed to study the safety and efficacy in this population. If you look at the natural history data in children and adolescents, a cohort of these patient population who had a visual acuity of 20/40 or better showed a rapid progression to legal blindness within 7 years. In contrast, patients in TEASE-3, and I'm showing you the data on 5 patients who completed 2 years plus, they showed a relatively stable visual acuity over several years. Each color here is representative of each patient, and each line is corresponding to each eye. The patients in this study over 2 years did not have any serious adverse events. Adverse events were of very mild nature. And once again, we did not see any adverse events related to vitamin A deficiencies in the visual cycle. TEASE-2 is a double-blind placebo-controlled study in patients with intermediate Stargardt disease, and the data from this will be presented in an upcoming congresses. AMD and Stargardt disease share a common pathophysiology of increased accumulation and formation of vitamin A dimers. Therefore, we studied in this population as well. It was a double-masked placebo-controlled study of 24 months in patients with a baseline geographic atrophy. The primary endpoint of this study of growth over 2 years showed a meaningful reduction of 13.4%, did not meet the statistical significance, but a sensitivity analysis of 6 to 24 months showed an annual reduction of 15.3 with a significant p-value. On the right-hand side, you will see the gildaretinol showing the LLVA 4.4, fewer letters lost with the gildaretinol. Overall, the safety is very well studied in this population. 400 patients have been exposed thus far, and we did not see any evidence of visual cycle deprivation of vitamin A, and no significant safety signals have been seen. With that, I just finished, and happy to take any questions if there is some time left.